Hello, my name is Eva, and today we're going to be talking about some tips and tricks on how to implement COLOREX orientation media. Step 1. Get familiar with orientation media. Inoculate a few sample plates with well-characterized strains. Suggested species to inoculate are as follows: E. coli, Klebsiella, Enterococcus, Group B Streptococcus, and Staph saprophyticus. Then inoculate 5 to 10 sample plates with urine samples confirmed to be positive for urinary tract infections. Finally, incubate aerobically at 35 to 37 degrees Celsius and read the next day. Step 2. Evaluate 200 to 500 urine samples with orientation media. Inoculate 20 to 50 COLOREX orientation plates per day in parallel to the plate set already in use by the laboratory. Most laboratories will be transitioning from blood and McConkey agar. Plates can be read the following day after proper incubation. For this portion of the evaluation, we recommend utilizing 2 techs to alternate between plate types every day. For example, on the first day of the evaluation, Tech 1 will read the orientation plates and Tech 2 will read the laboratory's plate set. On the second day of the evaluation, they should switch, whereby Tech 2 would read the orientation plates and Tech 1 would read the current plate set. This pattern should continue throughout the entirety of the evaluation to eliminate any technician bias. Step 3. Identification and AST. For orientation media, confirm 10 colonies each day from different plates via MALDI-TOF, then send to AST. Use the SOP already in place for the laboratory's current plate set. Step 4. Record the results. Record all data in the provided spreadsheet. Please reach out to us if you are in need of a copy for your own evaluation purposes. If you are able, please also take pictures throughout the evaluation. Step 5. Contact us. After the evaluation is complete and your results have been recorded, please reach out to us to discuss the data, pose any questions that you have, and talk about the next steps. Now that we've gone over steps for implementing orientation media, we're going to move on to some frequently asked questions and answers. Question 1. Can you do biochemical confirmation tests and MALDI ID straight from the orientation plate? Yes. Published scientific studies have shown that you can transfer colonies directly to MALDI-TOF from the orientation plate without any interference. Question 2. Is this media confirmatory? This media is 99.8% specific for E. coli, allowing for direct transfer to AST without the need for MALDI confirmation in between. All other colonies are presumptive and should be confirmed via MALDI before transferring to AST. Question 3. Can you perform AST directly from orientation media? Yes, for E. coli colonies. Question 4. After implementation of orientation media, do you still need to use McConkey agar in addition to orientation media for urine cultures? No. Orientation media detects both gram-negative and gram-positive bacteria, allowing for all common UTI pathogens to grow and be visualized on the agar. Some laboratories prefer to still include a blood agar plate, however, this is also not necessary. Question 5. Does Proteus swarm on this media? No. Orientation media is electrolyte deficient, which prevents the swarming of Proteus. It will have an isolated appearance on orientation media, allowing for other growth to be clearly visualized. Question 6. Can Group B strep be detected on orientation media? Yes. Group B strep appears as light blue-green translucent pinpoint colonies which are clearly distinguishable from Enterococcus, which appear a darker turquoise blue color. If you have any further questions or would like to start implementing orientation media into your own laboratory, please feel free to reach out to us. Thank you for watching.